My dad was diagnosed with ALS when I was about 9 years old. So you don't get signals to your muscles and therefore they become useless progressively.
The grieving process of, thinking all the things that he's gonna miss out, his son's sports, doing things with him.
He's not gonna be able to do that. I'm sorry.
Please welcome Hui Kai, Program Chair, Vice President and Head of Content, Wuxi AppTech, to kick off the online event.
Good morning, good afternoon, and good evening, everyone. It's my pleasure to welcome you to the 9th episode of the Wuxi AppTech Rare Disease Webinar Series, Collaborations that Transform. Today, we focus on ALS and Neuromuscular Disorders in honor of the World ALS MND Awareness Day. Neuromuscular disorders primarily affect the nerves that control voluntary muscles. These disorders result in muscle weakness and fatigue that progress over time. Most patients eventually lose the ability to walk, to dress, to speak, to swallow, and to breathe. Among the numerous neuromuscular disorders, ALS is perhaps the most recognized one, thanks to the Ice Bucket Challenge. Millions of families are suffering from these devastating rare diseases. Unfortunately, there is still no cure. Today, more than 20 distinguished speakers from global patient groups, top medical institutions, and the healthcare industry will join together for this global online event. We will center discussions on scientific insights, new approaches, and new technologies that will enable us to move the needle on ALS and neuromuscular disorders so that we can bring better medicines faster to patients. Together, we hope to raise awareness about these devastating rare diseases, to inspire new thinking that can advance scientific breakthroughs, to strengthen the global collaborations among us, and to empower people affected by ALS and neuromuscular disorders. Thank you for joining us today. Let's tune in now.
Please welcome Kalanit Balas, President and CEO, the ALS Association.
Thank you for having me today. My name is Kalanit Balas, and I have the honor and the privilege of being the President and CEO of the ALS Association here in the United States. I also have another honor, which is I happen to be the chair of the International Alliance for ALS and MND organizations around the world. And with my experience here, the passion and importance of innovation could not be more clear. At the ALS Association, what we are trying to do is make ALS a livable disease. And that means proving and pushing technology and therapies far and fast and in a really accelerated pace to make sure that people living with ALS can live longer, better, higher quality of life in the way that they want to show up every day. And there's a really good example I wanted to share with you today, which, if you remember, just a few years ago, there was an international global phenomenon called the Ice Bucket Challenge. Recently, Canada, Health Canada, has approved one of the first drugs that was ever funded from that Ice Bucket Challenge back in 2016. That is amazing progress as we look towards how do we make ALS livable in an incremental space and hopefully at a very accelerated rate as we look at new therapies and new technologies. So from the bottom of my heart, I thank you for all the work that you do, for your piece in this very complicated puzzle. And I hope you enjoy the conference and all the information that's coming your way. Thank you again.
Please welcome Lin Kaui, Founding President, Motor Neuron Disease Association Singapore, whose speech will be read by Muslim Al-Bakri, President of Motor Neuron Disease Association Singapore.
9 years ago, I had not heard of ALS or MND, but I knew Stephen Hawking. Now, I am a late-stage patient. I have a feeding tube and tracheostomy to sustain my physical body, and I depend on eye gaze technology to communicate. I am able to speak, though not clearly, which is still most wonderful. The Singapore MND Association was formed 14 months ago. Since then, MNDA's focus has been on the outreach, awareness, and advocacy programs. We have active Facebook postings, a thriving website, and chat group. We patients and families constantly encourage ourselves daily on these platforms because only patients understand what and how we patients are living through this terminal illness daily. Our persistent emotional struggles, our constant physical discomfort, and our burden on our families is very hard for any person to even imagine. Luckily, the minds of most patients are still very good, which is a double-edged sword, however. It is difficult to have a positive mind trapped in a dying body. The MNDA also organizes regularly online sessions to share information and for interaction amongst members. We members even had a picnic on the beach. This made everyone excited and happy. Yes, we patients can live our lives meaningfully, with dignity, receiving care and sustaining hope. Singapore in recent years has made good progress in the areas of medical care and counselling for patients. There is a dedicated medical team visiting patients at their homes, patients who need ventilation and respiratory support. I am one of the 200 plus lucky home patients. We patients need good news to sustain our hope, to give us reasons to live on. We want monthly updates on all the various research being carried out. Within 2 years, we patients want to see the degeneration stop. Within 5 years, we patients want to see lost function, especially breathing, swallowing and mobility, come back to us. Within 8 years, we patients will help in our community work to others. And within 10 years, ALS and MND will be history. I hope to fulfill my promise to my baby granddaughter, which is to walk her down the aisle during her wedding.
Neuromuscular disorders include a wide range of diseases, with hundreds of genes implicated in the pathogenesis. Right now, our key priorities are ensuring there are treatment options to improve symptoms, halting disease progression, and enhancing quality of life. What are the challenges that we are facing? How can we work together to better understand the genetic causes of neuromuscular disorders and drive technological innovation to benefit patients? Please welcome Karsten Baumann, Chief, Neuromuscular and Neurogenetic Disorders of Childhood Section, Senior Investigator and Acting Chief, Neurogenetics Branch, NNDS, NIH. Matthew Harms, Associate Professor of Neurology, Institute for Genomic Medicine, Columbia University, and Sharon Hester-Lee, Chief Research Officer, Muscular Dystrophy Association.
Well, so thank you everyone for joining us on this series on rare disease. And specifically, we're going to talk about moving the needle in ALS and other neuromuscular disorders. So the talks that you will hear later will be focused in on ALS, but we're starting right now from a very broad, big picture look at neuromuscular disease as a whole and how ALS and SMA and Duchenne muscular dystrophy and myasthenia gravis and all this broad array of diseases actually fit together in a category. And I think if you think about neuromuscular disease now and the huge breadth of them, it may not seem like they all have a lot in common, but if you put yourselves in the place of physicians a couple of 100 years ago, we're seeing patients, and the one thing that these patients have in common usually is muscle weakness, and it's often progressive muscle weakness. And so the group of physicians studying these diseases tend to see all of these people together, and they really didn't know if this was an acquired disease or a genetic disease, if it was neurological in origin or a muscle disease, or even secondary to something completely different. And where we have sort of fallen out in the field is that organizations like the Muscular Dystrophy Association or the AFM in France, TRINMD in Europe, cover this broad range of neuromuscular diseases that include diseases that cause muscle weakness either because of primary underlying muscle problems or something related to the nervous system that controls voluntary muscle movement, or that could be central or peripheral. And so you end up with this broad group of diseases that are different. We know they're pretty different now in their origins, but a lot of times the clinical signs and symptoms are kind of similar. So I'll just stop there, and maybe if we will take a moment, and I'll have my guest, Dr. Karsten Bonneman and Dr. Matt Harms, introduce themselves a little bit further and talk just a wee bit about what your background is and your research interest is in this field to start. So Dr. Bonneman, if you wanted to start.
Yeah, hello. I'm Karsten Bonneman. I'm a pediatric neurologist and neurogeneticist. So I got into the neuromuscular field really 30 years ago, almost 30 years ago, after finishing pediatric neurology. And I was interested in the genetic basis, genetic causation of neurological disease. And after my training completed in Boston, I was looking for the right lab to follow that. And it was Luke Kunkel's lab that took me in. And Luke Kunkel, of course, is known to probably most of you as the person who discovered, along with other people, the dystrophin gene, cause of Duchenne muscular dystrophy, which, at the time was a huge advancement in science. He identified this gene not knowing its function just by its genomic location. So that really started the era of genetic genomic medicine in neuromuscular disease. And so I joined his lab and got into neuromuscular that way and never left. And my research interest is really in early onset neuromuscular disease, pediatric neuromuscular disease, things that are often present when the baby is born with it. And more we are moving towards not only understanding the genetic basis of these disorders, but also thinking about the appropriate therapies that result from these findings. So, give you an example. When I was in Lou's lab, and he had discovered the dystrophin gene, and I was involved in finding some of the early LGMD genes in his lab, and now we're almost. This was the mid-90s, so we're kind of almost 30 years later, and both Duchenne muscular dystrophy as well as the LGMDs that I was working on, beta-sarcoglycan in particular, are now in gene therapy trials in patients actively being dosed. So that gives you an example or view how the field has advanced. For me personally, when I started out, really the focus was on finding those genes, and it took years to find a single gene. We're all proud to find a gene in a single disease, and that has now changed. Finding a gene or a mutation really brings with it the responsibility to deliver a treatment as well. So finding the genes and the mutations are now the beginning of a therapeutic journey for that. It's not the end of the journey, it's now the beginning of the journey. So that is really the paradigm shift that happened over the last years. For instance, we discovered a pediatric ALS gene just recently, SPT-LC1, and with it, in the same paper, was a precision medicine approach to that disease. We discovered a rare mutation in a congenital muscular dystrophy that was hidden to us before because we couldn't look in the deep intronic part of the genes. And finding that mutation inducing a pseudoexon delivered a precision treatment right with it. And so that is a paradigm shift that I've been experiencing as a neuromuscular geneticist.
That's great. Yeah. I mean, I know we have over 300 genetic causes of neuromuscular disease that we're tracking, and that number expands, it seems, weekly. Dr. Harms, you want to talk a little bit about your background and interest in research?
Yeah, I'd be happy to. I'm Matthew Harms, and I'm a professor of neurology at Columbia University in the Institute of Genomic Medicine. So like Dr. Bonneman, I got interested in neuromuscular disease and uncovering the genetic basis. And like he just elaborated, really doing that as the beginning of a journey to then find some sort of a precision medicine approach that might allow us to zero in on a molecular therapy or a specific pathway or target in order to develop a drug or some sort of treatment that would undo or at least halt the neuromuscular weakness that was occurring. I started off by doing research uncovering the genetic explanations for limb-girdle muscular dystrophy and for spinal muscular atrophies. But then as the cost of sequencing got cheaper, realized that it was now amenable for more common neuromuscular disorders like ALS. And so I have, since 2015, been focusing much of my time uncovering new genetic explanations for ALS and trying to understand how it is that the genetic mutations that we do find then lead to neurodegeneration. And does that give us additional clues for pathways that should be targeted for therapeutic development? Sharon, to your point earlier about lumping patients together into neuromuscular disease because of the progressive weakness, I'm kind of I guess a poster physician, if there is such a thing, for that, that lumping, because I see patients in the Pediatric Muscular Dystrophy Association clinic, the Adult Muscular Dystrophy Association clinics, and then the ALS center clinics as well. So I see kind of the whole spectrum of neuromuscular disorders and the whole genetic and lifespan of people with neuromuscular disease. So I think that, I'm very comfortable with this grouping of patients. And I would kind of point out that in addition to just the historical manifestations that patients come, there are some very strong rationale for keeping this group of patients together. And that comes from the clinical trial outcomes assessments. So, for many human diseases, it's very straightforward. You're looking at a lab test that you want to reverse or an imaging finding that you want to prevent or see go the other direction. But for many of these neuromuscular disorders with progressive weakness as the outcome, you need carefully trained physical therapists and clinical trial outcome measurements that require a lot of training and are similar across those groups. So when it comes to kind of developing clinical trial infrastructure, I think there's also a strong rationale for many of these diseases kind of being held together in a group.
Yeah, no, that certainly makes sense to us. So at MDA, we have a patient registry called the MOVER patient database, and we cover 7 different neuromuscular diseases in that registry. And it's everything from ALS, SMA, Pompe disease, which is a metabolic myopathy, to limb-girdle, the dystrophies, limb-girdle, muscular dystrophy, Becker muscular dystrophy, Duchenne muscular dystrophy. And although these diseases appear somewhat different in their cause, the case report forms for the data that we collected actually have a lot of common core data elements across all of these disorders, because they tend to have some similar effects, say on respiratory progression. A lot of them have cardiac manifestations that are similar. So certainly we see the commonalities there, and we have a lot of clinicians in our network who treat people in all of these different areas of neuromuscular disease. And so we touched just a little bit, I think both of you have mentioned the large number of genes that have been discovered in these largely genetic diseases. So when we think about where the field has come, that's one obvious difference, is we have a much better understanding of the genetic causes of the disease. Are there other things that jump out to you of the way that field has changed since you first entered the field, or things that you think are notable? I'll just throw that out to anyone who wants to jump in there.
Yeah, I would argue that it is not even just the genes that we're now discovering. It's actually individual mutations within genes that are coming to the forefront. And so I think we're moving post this single gene, if you will, era into single mutation era, when we try to understand what they do mechanistically. So in the same gene, we can have different mutations causing completely different phenotypes. And also single mutations may have different levels of actionability, how you can address them on the molecular level. Some, as I mentioned before, for instance, with the pseudoexon, so a splice modulating therapy can be tailor-made to a particular given mutation within a gene. And the type of mutation then is more important than the gene, actually, and when you think about the treatments. So we are adjusting our molecular toolbox to what we find genetically, and that now drills down to the single mutation level. The other thing I think that is now moving into the forefront is the long realization that a single gene disorder is not a single gene disorder. Every single gene disorder is modified by genetic background, by additional genetic variants that play out in a single human being's genome. And that may be related to the primary disease, but it may also be related to the response to therapy. So, for instance, is a therapy toxic for one patient and not for another? That probably depends on the genetic background of that patient. So the genetic complexity really goes down to single gene disorders as well as response to therapies. And then, of course, and Matt is much better suited to talk about that, there's a continuum from single genes to susceptibility, from single genetic causation to complex genetic causation. And ALS may be a prime example for that, to understand it. And I think what we're realizing in the rare disease field is, yes, that may be the case, but studying a single gene disorder in the sense that it is a primary gene that causes it with all the complexity coming behind it, as I mentioned before, really is informing us to also address more complex multi-genetic diseases. So I think the time spent in addressing single gene disorders is time well spent to understand more complex disorders as well. Matt.
No, that's a very good point. As we've developed the technology and the speed to be able to sequence large panels of potentially causative genes for ALS patients, now in some places we're approaching comprehensive sequencing for entire ALS clinic populations. So like at the Columbia Clinic, every patient, gets a broad panel looked at. And so we are appreciating individuals who have, what people think of as kind of driver mutations, some of these monogenic explanations, which we now realize are not completely penetrant. So you can carry the gene mutation, but not everyone with the gene mutation goes on to develop the disease. And so can we look for and find those other modifiers that are leading to the expression of that gene in an individual patient with motor neuron degeneration? And by uncovering those modifiers, would we be able to impact the likelihood of developing that disease? We're also, now getting to the point with close to 20,000 ALS genomes or exomes where we can start to look for patterns in what those modifiers might be, or look for those other kind of polygenic explanations for ALS, which I think will kind of lead to additional therapy ideas where you might be tackling multiple different pathways at once in a given patient based on the complex genetics that you were just alluding to. I was going to say the other kind of emerging driver of our ability to even talk about molecular, each mutation individually and understand each mutation individually, since they are so rare by themselves, is kind of the international collaboration and kind of the breakdown of silos between different research groups that are working on these things. So while, my project, Genomic Translation for ALS Care, has looked at 1,800 genomes, and Project Mine has looked at 6,000, and there are other groups that have looked at 2,000 or 3,000 here. The fact of the matter is that one major change that's occurred since I entered the field is that now that data gets shared relatively quickly for overlapping information and for additional discovery that couldn't have happened when people were still operating in their own research silos. And so that's one additional thing that I think has shifted over the course of the last decade that is really empowering kind of this next round of discoveries for neuromuscular disease.
Yes, and that actually it could be more extreme and say this is mandatory for us to move forward in the field of developing understanding and therapies for these disorders. And so it's so important for gene discovery and gene understanding, as you pointed out, but it's equally important for understanding therapies and developing therapies and therapeutic responses. Because every single rare genetic disease cause of, for instance, a form of ALS or other neuromuscular disease may be small n's in terms of patients with that particular mutations, but every learning of a therapeutic approach in that patient group can inform a much broader group of patients and accelerate development of therapies down the line if the data is shared, if the data is made to be known. And that goes for the therapeutic approach used, the biomarkers used, the safety events that happened in it, the management of safety events, and the charting of outcome measures. I think every single rare disease, even small ends, can propel the entire field, because every single one of these therapies makes broader points that are applicable across the field. And I think because of the commonalities that we have identified already in neuromuscular disease, neuromuscular is particularly powerful to benefit from that approach if we put it into action. And I think we're seeing a very, very promising moves that direction.
Yeah, so since I've been in the field, which was over 20 years ago when I started in the space, there are now almost 15 drugs approved for different neuromuscular diseases that are FDA-approved drugs. So I think one huge shift I've seen is as we've understood the science better, it's been driving the technological development and driving everything that comes with it. So we've gone from a few companies in this space to several hundred companies who have things in the pipeline or drugs approved in the neuromuscular space. So that's created a lot of activity. Certainly has changed the environment quite a bit, mostly for good, I think. And another thing that I've noticed over time too is that the patient organizations in this space have become just more and more sophisticated. And the same thing that you mentioned about silos and collaborations we're seeing on the patient side as well. As these organizations mature, we've seen a lot more collaborative efforts, sharing of resources and information. And again, these groups, which are allied closely with the patient community and with the researchers, often are sort of the glue that pulls a lot of the different pieces of this research industry, different payers, even government pulls all of this together. It's sort of centered around patients themselves and patient advocacy organizations. So it feels like the field has matured quite a bit. We have, drugs approved. We have endpoints that have been used for drug approval, and that sort of paves the way for the next round of new therapies. And to use that as a segue, what is most exciting to you right now in the field? Maybe if you want to say a few words about what you're excited about seeing developing.
Yeah, let me start with some excitement, and then also maybe with some challenges that the field is also experiencing at this point and has to face. The excitement for me, of course, as a neurogeneticist, is that we now have therapeutic options that move ever closer to the genetic causation of a disease. And I think if you think about genetic disease, it's a tough target to treat. It's a tough thing to treat, a often progressive disorder that affects such vital functions of the body. And really, your effect of your therapy needs to be as good as it possibly can be to really make a difference in the life of a patient. And the promise, of course, of gene-directed therapies is that we are moving closer and closer to the origin of the disease, and hence expect the impact to be greater. And that, of course, only works if we get there with our therapies, and we get there effectively. And when I say therapies, I mean the entire gamut of molecular therapies that are now on the table, from gene therapy, gene replacement therapy, to base editing, gene editing, transcript modulation. All of these are therapies that are gene and transcript directed, but they need to get there. And so this is one of the challenges that we face in the neuromuscular field. One is get to the target, get access to the target. So that's important in ALS, for instance, or disorders that are nerve-based, where you have to get to CNS origin of the disease with the therapies. And then on the muscle side, the pure size of the target, the size of muscle as a body organ is huge. And so we have to really, in that, cover the entire diversity of muscles that are important for movement, respiration, cardiac health, and life, essentially. And that brings with it distribution challenges and also safety challenges, of course, with the doses that have to be used to reach every single of these muscles. And so those are the target challenges. And the other challenge, of course, is the frequent progression that we see in many of our neuromuscular disorders. So we have to get there and treat effectively while there is still cells and tissue to rescue. So we have a window of opportunity that demands early treatment, early recognition, and early intervention.
I think, Sharon, I would say that I'm most excited, and this is a little bit different between kind of the genetic muscle diseases and ALS, and that is in ALS, 5 to 10% of ALS patients have an easily identifiable driver gene mutation, and in some populations that might even be up as high as 13 or 14%. But there's this large number of individuals that we don't yet have the ability to identify kind of a key monogenic explanation for their disease. But by studying, the 24 or so genes that have been implicated as causing ALS, we've been able to zero in on some key molecular pathways that could be targets for all of ALS. So when you study what's happening and going wrong in patients with C9orf72, you can see some downstream pathways that are disrupted that are shared with individuals with ALS that don't have an identifiable gene mutation in C9orf72. And so one of the things that I think has been very powerful in ALS that I'm excited about is that as we've looked at the implications of these driver mutations, we've zeroed in on particular pathways that are disrupted in essentially all of ALS and can go after those as targets. So I'm excited about molecular therapies that are targeting the autophagy pathways, or even the unfolded protein responses, the way cells deal with protein homeostasis and misfolded protein and aggregation, the way that RNA missplicing might be taking place when TDP43 function is not present in the cell nucleus, for example, and the way that that's being reflected in misspliced downstream targets that look amenable to potential therapies. And so that's kind of where I'm most excited, even as we don't have great driver mutation explanations for the vast majority of patients. And in fact, maybe we won't, but we've gotten really great insight into these shared molecular pathways within motor neurons and related cells that are key for the entire disease. So the kind of the springboard from the genetic explanations out into the rest of ALS is what I'm excited about.
Yeah, and that's a really powerful argument for identifying genetic causes and then understanding more sporadic disease. But also the complexity of genetic causation of ALS tells us that there probably is also complexity in sporadic ALS in terms of what an individual patient is a major driver amongst all of the downstream consequences. And I think the future, even here, will be precision medicine to understand this patient has this particular aspect of ALS pathogenesis as a major driver, and therefore we tailor make a treatment that addresses that predominantly, whereas another patient may have a different driver that is driving, and then we use a different therapy to address that driver, even in sporadic ALS. So I think the challenge here really is to continue to collect all of this data and all of the patients and really understand subcategories, understand individual drivers in all of these seemingly similar disorders.
Which raises the very important point of kind of figuring out biological markers of those different subtypes of ALS, right? And getting at, fluid and tissue biomarkers that might tell you, is this an ALS patient for whom neuroinflammation is the key driver, or for whom it's, purely a protein homeostasis challenge, to be able to kind of put patients into those different biologically similar groupings for clinical trials, for gene discovery, for mechanistic understanding, and what have you. And so, I think one of the other things that I'm very excited about in the field are all of the longitudinal biofluid biosample repositories that have sprung up, the natural history studies that are trying to link those biofluid changes to disease progression and disease manifestations. And even, one of the ways that we're going about that is by studying pre-symptomatic carriers of these gene mutations, sometimes even decades before we anticipate that they may develop symptoms, to see what are these biofluid changes, imaging changes that might herald symptom conversion that might give us a handle on some of those earliest changes that take place, that we could then, go and test in people who don't have genetic mutations to see if they're similarly occurring in advance of disease or at the time of symptom
and
conversion.
that gives you really the opportunity and hopefully a window into early treatment, perhaps even pre-symptomatic treatment, if your biomarkers are strong enough predicting a later manifestation of the disease. That would be, of course, wonderful if that materializes. But the other important, I think, of biomarkers that you mentioned and longitudinal following in these diseases, biochemically and clinically, is, of course, clinical trial design and clinical trial efficacy. So the more precise we are about the causation, the more precise we can be about the biomarker, and the more tightly we can design clinical trials that actually give us an answer whether an intervention is working or not. Because with so many causes, so many therapeutic avenues, and such limited resources, we need to be efficient in clinical trials and really doing what's necessary to tell us this is working and this is not working.
Yeah, you mentioned clinical trial efficiency. I think never has that been more important. As these many pharmaceutical companies are developing drugs for neuromuscular indications and wanting to bring them into clinical trials, is kind of the challenge on the clinician and clinical trial infrastructure that currently exists in most places. I think every center I know is looking to hire additional neuromuscularly trained clinicians to be able to help with the exciting clinical trials that are coming down the pipeline. We're all struggling to train enough clinical trial outcome measurement folks and get them as skilled as possible to be able to record clinical trials. And so I think as people are thinking about developing clinical drugs for clinical trials, we also need to be thinking about capacity within our neuromuscular centers to be able to execute on those clinical trials. And that's something that I know many centers are facing as a challenge right now, just needing well-trained research individuals to come on board and join us in this very exciting mission and at this very exciting time.
Yeah.
Great opportunity for trial networks and collaboration across sites. Mm-hmm.
Yeah, we're basically at a very exciting time where things are growing rapidly and our knowledge is increasing and we're making real progress. I think that's being felt by the patient community. But they're growing pains. And I think that's what we're putting our fingers on now, is the need to make sure that we are prepared to succeed by having all the resources that we need in place. And I think it's a good problem to have. It's definitely better, than we were 20 years ago when we weren't sure what caused most of these diseases. So it's remarkable progress. It's a very exciting time. And I thank you both for this discussion, and hopefully you will enjoy the subsequent talks that will be focused on ALS. So thank you all.
Thanks, Sharon.
Thank you.
With the global population aging rapidly, the number of ALS cases worldwide could increase from 222,000 in 2015 to more than 376,000 in 2040, representing an increase of 69%. With no cure and no effective treatment to halt or reverse the progression of this disease, the world is working together to fight against ALS, bringing a wealth of new scientific understanding about the physiology of ALS. What are the innovative targets and pathways that researchers are going after? Why it is important to seek a multidisciplinary approach for ALS treatment? What will the landscape of ALS research and care look like in the near future? In this session, our guest speakers will share their insights. Please welcome Jeffrey Rothstein, Director of Brain Science Institute, Director, Robert Packard Center for ALS Research, Johns Hopkins University, Sabrina Paganoni, Co-Director, Neurological Research Institute at Massachusetts General Hospital, Physician Scientist, Healy and AMG Center for ALS, and Kuldeep Dave, SVP of Research, the ALS Association.
Well, thank you for the WuXi AppTech team for inviting me and Jeff and Sabrina to be on this panel. Maybe let's start with better understanding of the biology. Jeff, Sabrina, this is what we like to call target ID and target validation. And in the past, target ID, target validation was let's look at cells, let's look at animal models. But now we have tools that are much more human relevant. And so, Jeff, would love to hear your thoughts about, the use of some of these human models to optimize, to make target ID and target validation better.
Yeah, so I agree with you. I think there are newer models. I mean, we spent 2 decades using rodent and fish, and to be clear, these are they have been useful tools, but they're not human. And I think our patients certainly all sort of point out that we haven't as many drugs as we'd like, and why are we using mice? And fortunately, the technology has increased in the last decade. Now we actually do have human models. We have what cancers had for a long time. We have the way of essentially biopsying the nervous system by using blood cells from patients and creating IPS cells, induced pluripotent cells. And these are then turned into our patients' cortical neurons or spinal neurons. And those, some, not all, but many of us believe is sort of a future platform. When you think 10 years in advance, is this the platform that'll teach us more about what's really happening in patients? Sure, it's not a connected brain, but on the other hand, most of our drugs aren't about connected brains, like a disease like epilepsy. They're about what's wrong in a given cell and the cascade of events that goes wrong in a cell. And so these cell paradigms, these new cell paradigms are really beginning to be used worldwide. The other really important thing about this technology that trumps, in my mind, really many other approaches is that many of the models we've used for the past 25 years have relied on the mutations of ALS, which we know are really important to teach us how the disease begins in that subset of patients. But they don't necessarily teach us what's going on in the vast majority of our patients, sporadic ALS. And since these cells can be built from all of our patients, in fact, they have, one of the programs I'm involved in, Answer ALS, but there are others around the world where we derive these, I tend to call them biopsies, from our sporadic patients. They may give, and I want to underscore may, because I don't know for a fact that they'll be useful, but what I do know is that the mouse has not been as useful as we've hoped. And these will give us a platform that's human-based, that's really based on sporadic ALS. And when I think of the next 10 years, I really think that's what we have to focus on as much as we can, the real population of ALS patients, sporadic patients. And to be clear, it's not the only model system. People are actually taking the cells and sort of rebuilding forms of the brain called organoids, another way of looking at cell-cell interactions, because we know in ALS that's not just the neuron that dies, that there's inflammatory cells called microglia. There's other support cells, glial cells, oligodendroglia and astroglia. And all of these human platforms may give us that window into 2 new ways. I don't want to lecture, so I could stop, but I think that's important.
Yeah, and Jeff, what I'm thinking about is target ID and target validation were really slow. And what I'm hoping that in 10 years is we are quickly able to nominate the targets, validate them or invalidate them, and quickly close the door on them. Because, the thing I see in the field, and Sabrina, I'll come to you in a minute here, but, is that it's taking time. And so 10 years from now, if we can use these human systems that you just mentioned, Jeff, to, quickly say, is this target druggable? Is this something we can take into the therapeutic pipeline? And answer that question quickly. I think speed matters. We got to work on the ALS clock, and so, that is my hope for the future. Sabrina, coming to you, there's this whole idea about even using patients' own cells as a personalized medicine approach, and would love to hear your thoughts on, as a clinician, how do you see the promise of that, 10 years from now?
Yeah, that would be fantastic. That follows what Jeff was describing in terms of target validation, using cells or material that comes from actual patients, not just relying on specific mutations that can be tested in the mouse model, for example, but really giving us the opportunity to test drugs that potentially can be personalized. I do want to mention that what I think is going to be really important in the future, again, thinking about what comes next, is that as we discover, hopefully, at least a few drugs that have a positive impact, it will be very important to go back to the models and then actually test them in the models and then validate the predictive value of the models, because the truth is that we still don't know which model, and obviously the devil is in the details, even when it comes to cells, personalized cells from patients. How do you actually, what's the best way to grow them? What's the best phenotype that you're looking for? So as we develop new drugs, and quite frankly, even with the ones that we have, it would be great to then create almost a battery of tests, we validate the ones that are most predictive, and that could be more than one actually, and that will actually speed up the drug development process.
That's a really good point, Sabrina. And the other thing I think about here is, at ALS Association, I get asked all the time, how many models should we test our drug in? And I always say, I'd rather you test it in one model that makes the most sense mechanistically as to how your drug works. So if your drug is reducing aggregation, then use a TDP-43 model instead of a SOD1 model, right? Or if your mechanism is targeting mitochondria or oxidative stress, then use the SOD1 model. But I'd rather you use one model and do a much more deeper validation in that one model. Well, do a full dose response. If it's an agonist, antagonist, do a pharmacology study, do PK, instead of trying to do efficacy in 2 or 3 different models. And so I'm so glad you talked about that. Jeff, what's, one of the things that you brought up was inflammation. And, the one thing that you and I have talked about this in the past is, right now, if you look at our ecosystem, 40-50% of drugs that are in the pipeline right now in phase 1, 2, and 3 are neuroinflammation focused. And, do you worry, like I worry, that, we may have all of our eggs in one basket? And do we need to expand more? And does our understanding from omics, from initiatives like Answer ALS, help us expand and diversify the pathways we're looking at?
So there's a lot to that, and I'm going to go through a couple of things quickly. One, so I'm the most gray-haired person here. Trials in the 90s were really when there was a growth of ALS trials, began with some of the growth factors. A huge number of trials in the 1990s were what we as neurologists, especially neuromuscular specialists, ALS is sort of only one of many of our neuromuscular diseases. Many of them were actually autoimmune diseases. And so we were pretty familiar with using anti-inflammatory related pathways. And so the 1990s was littered with trials around anti-inflammatory drugs. Obviously, they didn't work because we'd be using them, but to be really careful about that statement, the trials in the 1990s are not as sophisticated as they were today. So it could have been that we missed something. There is a huge number of drugs right now in trial that are based on, if you will, immune or inflammatory pathways. And part of it is we look at the pharmacopoeia of drugs available in all of medicine. There are a lot of drugs like that, and the drug companies are coming to us. The good side of that is there's a couple of good things. One, it's good to have trials. It's good for patients. Just being in a trial, and I'm sure Sabrina will talk about this in a moment, just being in a trial is actually generally good for patients. They do better in trials. So most of them fail. Of course they will. In medicine, 80% of all drug trials that are phase 2 positive fail. But it's an advance. We're doing so much better. We're building new biobanks for biomarkers. All of this in aggregate is very good for patients.
Right. And Jeff, one of the things we're doing at the association is also looking across our portfolio and seeing, do we need to do special call outs for mitochondrial based approaches or endoplasmic reticulum based approaches, right? Or lysosomal based approaches. And so this way we can, even through our funding, start to have an effect on the type of pathways that are being explored. Now that you brought up trials, let's maybe switch gears a little bit. Sabrina, I'll come to you on this is, how do we make trials faster and efficient? We just talked about faster target ID and target validation on the preclinical side. So on the clinical side, how do you do that? And, you have a blueprint that you have been co-leading around the Healy platform trial. So would love to hear, how do we think about faster and efficient trials?
Yeah, no, I'm happy to talk about that. I also was intrigued by many of the comments that Jeff made, and I agree with everything you said, and we could continue to develop that line of thought. But with relation to trials, I think that the most important part of a trial is that it should be designed to provide a decisive answer. So for example, if we're testing multiple drugs that work on inflammation or neuroinflammation, we should be able to, at the end of the trial, to either rule in or rule out a certain target or pathway. So I think that the problem I know, Jeff, you had a little bit of a history, you gave a little bit of an overview of, sort of the history of trials in ALS. And the problem with some of the initial trials was that, again, they wouldn't provide a decisive answer because maybe we didn't have the biomarker. We were not sure if the drug didn't work for one reason or another, not necessarily the target itself. And so now over time, and those were the first trials, and that's fine. When you start doing trials in any new field, really, there were no trials before, and now there were a few, and then we continue to perfect them. And I think that now the contemporary trials are more sophisticated because they can provide a definitive answer about specific drugs, but also specific pathways. And so by process of elimination, if you will, we can also rule out some aspects of for example, a certain class of drugs or subclasses, right? Inflammation is a big term, or autoimmune drugs are a big term. They actually have, there's many different subgroups there. So with respect to that, what I have been working on the clinical research side of things, in collaboration with many colleagues, is to make the trials better. Right now, we're testing 5 drugs, and we're adding a couple later this year. And so the idea is to create an infrastructure that includes those, best practices that as a field we have developed over the last few years, so that we can actually test drugs in an efficient way. And I would say that the platform trial itself, which is, drew inspiration from oncology, because certainly in cancer they are ahead in terms of the efficiency of trials, but we brought that concept to ALS. And now with the Healy ALS platform trial, we have this infrastructure that, again, includes many of the needs of the community, both in terms of the scientific community, collection of samples in a certain way, sharing of samples in a certain way, again, to spread the knowledge, acquire knowledge and also share it. And also a lot of other needs of the industry partners for robust trials that can provide a definitive answer, needs of the patients to have an accessible trial. So there's many aspects to that. So the trial itself cannot be a cookie cutter, right? We had to incorporate a lot of the knowledge that we accumulated over the years.
Yeah, and Sabrina, I really like what you said. It's not just faster and efficient, but decisive trials. We need to have an answer at the end of that trial. Did the drug work? Did the drug not work? So Jeff, this is one of your topics that you like to talk about quite a bit, about target engagement markers, knowing if you're actually testing the mechanism of your drug in the clinic. Would love to hear your thoughts about this.
Yeah, so again, a little quick history, and Sabrina's right, the trials in the 90s, even 2000, this has been a wonderful evolution of how we do clinical trials, because in the grand scheme of what we do in ALS, for that matter, any disease, if you have elegant preclinical science, laboratory science, and by the way, it's not always elegant, but let's assume it's elegant. If your trial, the ultimate experiment, is not well designed and well done, what a waste. You've got a great idea, you think you've got a great idea, but you do the human experiment and it's poorly done. And in retrospect, and it's really more of an evolution, the trials that we did a long time ago, they were small. A company would not always listen to us because it took time. And today, what Sabrina described to you is those trials, when companies come to us, we actually have a panel as part of NEALS, the Northeast ALS consortium that we have, where companies come to us and begin to describe what they want to do. And they're now asking our advice. And we sort of tell them, well, good idea, interesting drug, but you got to do X, Y, and Z. You got to figure out if it gets into the brain. And if you don't, you can do what you want. Now, that's not going to be a good trial, and you're going to probably waste money, and you're certainly not going to help patients the way we think an intellectually well-designed trial should be done. So target engagement is really, really important. One thing I'll point out, though, and second, and after that, so it binds its target, whatever it's supposed to do, and can you show that it's affected the disease cascade, something that's supposed to tell you it's mitigating the injury of the disease? That's really nice, too. We have early markers. We have something called neurofilament L. It tells us, well, when a cell dies, it releases this. Maybe we can slow that down. And on the horizon are real markers that teach us about that injury cascade. One's called TDP43. We can talk about that if we want, but it is one of those markers. This comes back to the very platform we started with in today's discussion, those iPS cells. They tell you not only a potential pathway, but you can test your drugs in these living cells from the same patients. And when you do that, not only can you show if the drug is working at its target engagement, you can actually have a readout to say, well, what's changing? And whatever changing, because that maybe I can measure that in a patient, their blood or cerebral spinal fluid through a spinal tap. So you get both the marker of drug efficacy as well as the evidence that it's working at its target. Yeah. So that's exciting for the future.
Yeah, maybe one quick thing, Jeff and Sabrina. We talk about trials and more trials, but more trials means we need more people in the trials. And how do we get more people in the trials? How do you both, as clinicians, talk to your people with ALS, patients who come into your clinic? How do you talk to them about genetic testing? That's not always an easy conversation to have.
Yeah, no, I mean, it's still a complex conversation. I think that what's most important for me as a clinician is to share the availability of targeted products or investigational products that could be applicable to that person should the test results be positive. So I think that that's really what has really changed over the last, let's say, couple of years. Because before then, quite frankly, it was still a complex decision, with no direct applicability. Right now, the situation has really changed over the last couple of years. And so I think that now the conversation is also about the possible access to investigational products that are in development for specific mutations. Now, it's not for all mutations, and some of them may work and others may not. So it's still at the experimental stage, but certainly, this change in terms of the availability now of targeted trials has really completely changed the conversation in my mind.
Yeah, I completely agree. And my conversation, first of all, every patient I see is now offered genetic testing. Obviously, it's up to them. I never, of course, force it. And then I divide it in a different way, the three different bins that Sabrina just mentioned. One, it's potentially diagnostic. It depends when a patient gets to our clinics, how early or late in the disease is. Secondly, it's exactly what Sabrina said too, which is that it makes them eligible for exciting trials, gene therapy. Not all, but if we don't know their gene mutation, if they have one or not, we need to know that for trials. And then lastly is the family issue. That's actually one, at least the way I practice, I often let them know there's a possibility of how it can be important to the family. But generally in my clinic, if they then want, maybe a spouse is there or a child's there, I bring in genetics counselors for that phase. I'm not a genetics counselor, but I can talk, the patient's my patient. I can deal with the initial issues, diagnosis and trials. And then we usually bring in a genetics counselor for the last bin. But the value of that is enormous. And to be fair, I don't know the exact number, but at least 80% of patients, maybe it's closer to almost 100%, now get these free panels. And the fact that they're free is fantastic. I can tell you years ago, they were a fortune, but it also means I'm finding genes that I would have never found before in patients. I'm finding patients who have mutations that account for their disease and just wasn't obvious. And that's really important. It's not important for them, for future drugs. And just to add to that, especially when it gets to family, as you know, and of course, Sabrina knows, at least some of the mutations, and now I'm speaking of the SOD1 mutation, we're already into the next generation of medicine, which is to say, someone carries a mutation, they're not ill yet, we're going to be following them. And as soon as there's a hint biologically, not even clinically, biologically, if there's something wrong, we could institute therapy. That's getting in early. And that's what one day we have to do with all of our patients, get in as early as possible. And it's genetics that's allowing us to do that.
Exactly. And, this is one way to also increase the number of people, the pool that's available for clinical trials, because as we know, later diagnosis means maybe they are now no longer, in the inclusion criteria. And so they can't, even if they want to, they can't participate. So I'm glad that you brought up that. So, maybe we switch gears, Sabrina and Jeff.
Before we do, Kuldip, I want to bring up one more thing. In terms of increasing patients, I literally just had this conversation the other day. We do have set criteria. We as a community, and of course, the whole point of a trial is, can we prove in the time frame of the trial that we've done something to slow the disease down? It often means we exclude a fair number of patients. Of course, Sabrina knows as well. But early trials where you're just trying to figure out, does the drug work at all at its target? This gets back to the target discussion. So not clinically, not but you need to first know before you even get to an efficacy trial, does it work at its target? Those patients don't have to be so restricted. You can take patients that, what we call slow patients, patients who have the disease for many years, who are not eligible usually for trials. But if we have those biomarkers, this gets back to the TP43, and we can show that there's a pathway in them. It's an injury pathway. Wow, you have a reporter for, does my drug affect that pathway? They may not show clinical efficacy, but your drug's affecting the pathway. And that opens the door to many, many more patients who at least could be in phase 1 trials. Yeah. So I would argue, I'm throwing this to Sabrina.
Yeah, and actually, I wanted to expand on that. I completely agree. I think that this also goes back to the point of collaboration between academia, the patient community, and industry. Sometimes trials can be designed as sort of cookie cutters, right? Like kind of, if that's always been done a certain way, you see how inclusion and exclusion criteria are, and they can be repeated. Instead, I think we have learned as an academic community to really be flexible and really use certain criteria for the right phase of development.
Well, thank you both. What I was going to say, something Jeff said earlier about, that interaction with the person with ALS and their family. I wanted to touch on sort of the care part of this, the managing ALS today. So we talked about drug discovery, and that's for, in the future, we'll have a drug, but there are people living with ALS today. And there are just so many decisions, as Jeff, you and Sabrina just talked about, that you all have to make in real time, looking at where the patient is in the disease journey, when to provide respiratory intervention, when to provide GI intervention. And so can you, Sabrina first, and then Jeff, can you talk about sort of the concept of a multidisciplinary clinic and why is that so important? How is that helpful for the outcomes for that patient?
Yeah, both Jeff and I work in multidisciplinary clinics, and at this time, there are several clinics. I think there should be more, but certainly there's many clinics in the US and globally that really specialize in the care of the person with ALS, and that it's always a collaborative approach because ALS affects many areas, obviously many regions in the body, and requires specialized expertise to really maximize outcomes. It's been shown many times that attending a multidisciplinary clinic improves outcomes. I would say that while I love clinical trials, and that's what I do most of the time, I never forget, and in fact, I always emphasize with my own patients I see in clinic, that we already have interventions that are proven to be effective and prolong life and survival, such as, for example, initiation of BiPAP when it's important to do so. And for sure, we have several interventions that can improve quality of life, quite frankly, much more than a drug that we don't know if it works yet. So, and the reason I emphasize that is because, unfortunately, sometimes, again, the importance of multidisciplinary care is not as emphasized. And so it's really, really critical that we give all our patients access to the best possible care, because otherwise, we're missing out on things that are already available and should be available to all.
Yeah, I couldn't say any better. That's so true. And the other point to this, and at least for me, if I'm honest, I'm a neurologist. I'm not a pulmonary physician. I'm not a nutrition specialist. So you need, and since patients don't have they come to us with neurologic problems, but the manifestation on their body are many, many systems. They can't eat, they can't take their proper nutrition, they can't breathe properly. And so we need those experts in different areas. In fact, I often tell my patients, fine, you've met me, we're going to talk about what's going on in you and make the diagnosis. And now I'm going to hand you to my team that's really going to keep you in the best shape possible, which are those other physicians and practitioners, occupational therapists, physical, everything that Sabrina just mentioned. And that is the best care. I often use the analogy with patients. I tend to use cars. Patients aren't trained in medicine. So we think of the Indy 500, you got this fantastic race car, but it's got to go to the pit crew where everyone really works on that car to keep it in the best shape. Our clinics are those pit crews to keep patients in absolutely the best shape possible.
Yeah, thank you both. And, I've been thinking about this in terms of the role of the multidisciplinary clinics is going to be even more important when a drug is actually approved. How do we, both from an infrastructure standpoint, but also from an operation standpoint, make sure that that clinic continues to provide better care, optimal care, while you have new drugs coming on the market? So this is really exciting. Jeff, were you going to say something?
Yeah, buried in what you just said is so right. Sabrina and I, big medical centers, some of the biggest in the U.S. that care for ALS, we're experienced, but there are a lot of patients out there, and what is lacking is that kind of care in the vast majority of the United States. And so one of the challenges in the future, especially if we have things like this, is when patients go back to, I'll say this, rural America, how do we make sure that they get that kind of care? Because they're not all going to be able to take 5-hour drives into their local city. And I don't have that answer, but that's something we certainly have to pay attention to. There's something buried in what I just said, though. There is something that's come out of the bad of COVID, and that's we all got used to telemedicine. We all got used to taking care of patients that couldn't always make it to clinics. Not necessarily, it's not, in my view, it's not the best for making initial diagnosis. You need hands-on, in my opinion. But once you have established that relationship and maintaining contact with patients, telemedicine is really quite important. And secondary, that's not really secondary, is the use of tools like apps, smartphone apps, and devices at home that can actually continue to measure patients' physiology, how they're doing, and feed that back to us, so they don't all have to drive hours to come to our clinics, but we stay in close contact with their true neurological status and then what we can do from afar.
Yeah, that's so important. And I think that will require, hopefully soon, if we have many drugs approved, some significant changes to the way healthcare is delivered for ALS. So I think there's going to be a need to better support and resource both the big centers that have already existing clinics, as well as their connection to maybe more remote areas where maybe it doesn't make sense to have an ALS clinic because the area is remote, but there should be an ability to connect with telemedicine or wearables or other tools, as you mentioned. I think this will certainly require some changes also in the reimbursement practices and sort of the way clinics are resourced.
Absolutely. Well, I think our time is coming to an end here, Sabrina and Jeff. Maybe just if all three of us could probably end on, sort of the optimistic, where do we see us in 10 years? I mean, I'll tell you what I'm really excited about. I hope I'm not stealing what you were both going to say, but we have never had this many ALS trials in the ecosystem. I mean, who knew for a rare disease like this? We're looking at, over 60, 70 trials worldwide between phase 1, 2, and 3. And that's a really exciting time. And what's exciting is that it's different type of approaches. So there's small molecules, there's antibody approaches, there's stem cell therapies, and there are different targets and different pathways that people are going after. So there are big companies and there are small companies. And so that's what makes me really excited. I think the concept of can we on the preclinical side validate fast? On the clinical side, can we test rapidly and in a way that informs how the drug works? And then on the care side, how do we make sure that when a drug is approved, that's incorporated into the clinic? And so that's what I'm excited about. And so, Sabrina, maybe any last comment for you?
Yeah, no, I completely agree. And I would say, and I'm sorry, Jeff, but I'm luckier than you, because I got into the field at the time of exponential growth. So you started it, and now I'm benefiting from that. And so, over the last 5-10 years, when I've been in this field, I myself have seen this exponential growth. So I'm very optimistic, I have to say, because even if I think about, actually, the other day I was looking at some of the initial slides. When I finished training 10 years ago, I mean, the field was so small, so much smaller, and few trials, and now it's really like exploded. So this is the right time for ALS. Yeah.
How can I not agree with all of that? So there's a couple other things. So one, no question, I actually believe in 10 years, we will have more drugs. I don't know how many, I don't know how powerful they'll be, but we will likely have more. I think we'll have a better understanding of targeting subgroups of patients, meaning sporadic patients. Or at least I'm hopeful of that with today's tools. The other thing buried in actually what Sabrina just said, and it's true. I mean, I have lived through a lot of that science from the first discovery of SOD1, the early mouse models, the early trials, the gene therapy trials. One of the big changes, it's not just that there are more companies, there's more of us. They're more intelligent, committed, both clinicians and scientists. That community is huge.
Well, thank you. I want to, again, thank the WuXi AppTec team for asking the three of us to be on this panel and share our thoughts about the future. We hope that in 10 years, this disease is a livable disease and that we have multiple treatments, that we don't have to even be afraid of telling someone that, you know, they are diagnosed with ALS, that they can live their life, albeit with treatments, but that, you know, that their lives are extended. So thank you to the team, and Sabrina and Jeff, thank you so much for joining me on the panel.
Thank you.
My pleasure. It's been great. Thank you.
Today, an increasing number of promising therapeutic approaches are in the pipeline, with potential to tackle the multimodal aspects of ALS and other neurologic diseases. What is the state of current and emerging pipeline? How are we addressing the complexity of biology? What about new targets and biomarkers? Please welcome Neil Cashman, CSO, Pro-MIS Neurosciences, Professor Emeritus, University of British Columbia; Michael McGrath, UCSF Emeritus Professor of Medicine, CSO and Founder, Navivo; Robert Scanneven, CSO, Verge Genomics; Fernando Vieira, CEO and CSO, ALS Therapy Development Institute; and Mulali Gopalakrishnan, Global Head, Neuroscience Search and Evaluation, Corporate Strategy Office, AbbVie.
I'll kick it off today. This is really an exciting time to be discussing on therapeutics pipeline in ALS, discussing actually and sharing the progress, particularly in a number of fronts that we'll cover during this webinar. First of all, there's been, over the past decade or so, increased understanding of disease biology and mechanisms in ALS. The field is coming up with very innovative and novel approaches for cellular and in vivo models for target discovery, validation, including utilization of some of the AI, ML type approaches to expedite early phase of drug discovery. There are pioneering new approaches for identifying the right patients and the right time with the use of biomarkers and translational strategies. And then there are a number of innovative therapeutic approaches that are in the pipeline that offers the potential to tackle the multimodal aspects of the disease. So these are a number of cardinal topics that we'll cover in this webinar today. We'll take a deeper look at all of these aspects in the coming minutes. So we're going to start off this session with Fernando, who's going to begin by outlining sort of the state of the emerging ALS therapeutics pipeline and some of the approaches that he and his team has taken recently. Fernando.
Thank you, Murali. And thank you, WuXi, for the opportunity to be a part of the panel today. It's a pleasure to be here. Like all of my fellow panelists today, I've been in the ALS drug discovery and drug development space for a while, more than 20 years. And the clinical landscape has, in some ways, it hasn't changed enough, right? Ultimately, we still have a critical unmet need for people living with ALS, but in a lot of ways, it's evolved dramatically. The clinical development pipeline is much, much richer than ever before. There are simply just more companies and more drugs being developed clinically in different phases of development and preclinically. And our understanding of the disease has evolved very much as well. It's really, and when I say understanding of the disease, really understanding of ALS as multiple diseases in some ways. And I think that's developed far beyond what at least I could have imagined, 15, 20 years ago. I never would have pictured the landscape that we have today. And I think that development has come from, it's been rooted in a few important advancements. I guess a lot of it is a better understanding of diverse genetic causes of ALS in particularly in familial ALS, that 10 to 15% of folks who develop the disease who directly inherit it from somebody in their family, from their parent. So we've learned a lot from that. And we also have a better collection of tools and technologies that can be brought to bear to better learn about the disease, both at a basic research level and also at a translational research level. So now we have a lot of companies and even nonprofit organizations like ours at the ALS Therapy Development Institute that have clinical development programs with treatments that target a whole gamut of possible approaches. So yes, we can go after specific genes in the case of monogenic associated ALS. But we also have therapeutic approaches that go at ALS as though it's a proteinopathy, focused on maybe, aggregates of SOD1 or aggregates of TDP43 or the C9orf72 dipeptide repeat protein. So that's one approach that we're seeing a lot of. There are approaches targeting, metabolic function, mitochondrial function. We see a lot of therapeutics going after immune dysregulation, seeing if there are ways to modulate those pathways in ALS. And even more recently, we're starting to see targets going after gene splicing dysregulation. So it's a really diverse collection of biologies that have been implicated in ALS. And there's solid evidence to support a lot of these approaches. They may all have some value and some validity. And the key challenge might end up being finding the right people and the right time in which to intervene in all of these specific ways. It may turn out that not all people will respond to all therapeutic approaches. So you kind of have to think about the Venn diagram and overlap between the folks with familial ALS and specific mutations and folks with sporadic ALS that may have these different pathways engaged in these different commonalities, which I know a lot of folks here will talk about today. I can talk a little bit about how ALS TDI is going at this.
That'll be helpful. Yeah, that'll be very helpful, Fernando. Yes.
Thank you, Marley. So I guess for us, our approach is we're a nonprofit, right? So we're a nonprofit biotech or research institute. And our focus is to try to discover and invent or help other organizations advance their own technologies or drugs for ALS. And then partner in a way that gets those into the for-profit sector and develop them clinically. What we do, the way we do it, we have a research lab here in the Boston area where we use a diverse collection of model systems, cell-based systems using induced pluripotent stem cells, other cell lines, and animal models, including zebrafish and mice, to really try to discover new drugs and optimize them through preclinical pharmacology, and then get them to an IND-enabled drug. And we're modality agnostic. So we have one example of a drug, a monoclonal antibody targeting CD40 ligand. So this is an immune target, which we've been able to partner and move into clinical development. Phase 2A trial just read out. It was good. It was promising. So that's one way that we've gone at it. But actually, our preclinical pipeline right now is focused on small molecule drugs, targeting mitochondrial energetics or post-translational modification type called protein arginine methyltransferases. So we think it's going to take a lot of different approaches, diverse modalities, diverse targets, to really ultimately address the needs of everybody living with ALS. And we're really excited about the idea of working with others to make that happen.
Thanks, Fernando. That's a very, very nice overview. And you did put the overarching aspects in perspective. So thank you very much for that. Just to continue on the same theme, and Fernando touched on the importance of targets and going after the right targets, mentioned about the genetically defined targets, in this case, the familial disease. Of course, the sporadic population is significant. And many of us know that the biology underlying TDP-43 is pretty fundamental to the overall pathophysiology of what's going on. So I want to maybe turn to Neil now to sort of elaborate a bit more on what's our understanding on TDP-43 biology. How are we tackling that aspect of the biology, either directly or indirectly, and share your perspective on, what are the challenges in this space as we explore the biology around TDP-43, which is a very fundamental component of the disease pathology. Neil.
Thanks, Murali. TDP-43 has been implicated in many diseases, especially ALS, but also frontotemporal dementia and some other neurodegenerative diseases like the late phenotype of Alzheimer's disease. TDP-43 is, we all understand what you and Fernando are saying, that there is quite a bit of heterogeneity in ALS, but we're not confined to treating each and every individual variant of ALS, because there are some features of all or virtually all ALS that are preserved. And one of those is TDP-43 aggregates in the cytoplasm. And this is interesting because TDP-43 also can have familial ALS mutations that lead to the disease, but the vast majority of TDP-43 aggregates are wild type protein that have not been mutated. So it may be kind of on the razor's edge for being involved in all or most cases of ALS. So the pathophysiology of TDP-43 basically has two facets. One is loss of function, because in ALS and in frontotemporal dementia, there is nuclear depletion of TDP-43. This eliminates TDP-43 from performing its normal functions, particularly important for neurons, which include RNA splicing. We might hear a bit later with Rob that Stathmin-2 and UNC13A are some of those proteins that have been implicated in ALS, which undergo splicing changes with the depletion of TDP-43, nuclear TDP-43 in disease. But there's also other losses of function. TDP-43, normal TDP-43, is engaged in RNA transport, stabilization, and even protein translation in distant compartments like the synapse or the dendrite. You can't knock down TDP-43 to avoid the cytoplasmic aggregates because of these vital, critical neuronal activities that it supports. Mm-hmm. So what are the gains of function? These are basically marked by cytoplasmic aggregation, which appears to induce inflammation via at least two pathways. And we'll hear from Mike about the role of inflammation in disease, but also promotes mitochondrial dysfunction, ER stress, proteasomal stress, translational stress with ribosomal collisions, the whole gamut. TDP-43 aggregates also have a surface that recruits co-aggregation of nuclear transport and pore proteins, alpha-synuclein, stress granule proteins, but it's a poorly recognized gain of function that may also produce the loss of function of depletion of nuclear TDP-43, and that's via its prion-like activity to recruit new copies of TDP-43 to the misfolded aggregate. So how do we tackle TDP-43, this pathology, the loss and gain of function? Well, directly, if we had a method or a drug that would break up aggregates and repatriate TDP-43 to the nucleus, that would be great.
Mm-hmm.
So that is a pathway that we're following in PROMISE Neurosciences to selectively target misfolded TDP-43 with antibodies and intrabodies. Indirectly, one can eliminate the negative consequences of loss of function of TDP-43. And we'll hear from Rob at Verge about iPSC technology to replace or allow stathmin-2 to be expressed to stabilize axons, and UNC13A to promote vesicle genesis in the nucleus. Robert will also talk about a kinase inhibitor, which is very important in the pathology of this loss of function.
Yep, that's very helpful. Very, very helpful. Maybe we could close with, what are the challenges in this space with TDP-43? Certainly an exciting target, but with respect to the challenges in targeting TDP-43, can you maybe make a closing comment on that?
Sure. So the indirect approaches are the most advanced, but it's unknown if correction of one pathway will lead to an amelioration of disease, although it is very hopeful. Mm-hmm. For our specific targeting of TDP-43, the challenges may include vectorization of intrabodies, which will be a gene therapeutic, which has really never been tried before.
Yep.
So these are the hurdles as I see them.
Great, thank you very much. And so Neil touched very much on sort of the role of the neuroimmune mechanisms in the overall pathophysiology. It's not unique to ALS, but I'd like to invite Mike McGrath to sort of elaborate more on the role of neuroimmune mechanisms in ALS, examples of targets and approaches, and what are the most advanced efforts in that space. Mike?
Yes. So many years ago, I started looking at neurologic disease, neuroinflammatory disease, by looking at AIDS dementia. There's a subset of AIDS dementia patients who had sort of classic ALS kind of symptoms. When the effective therapy for AIDS came along, I said, I'm going to work on a nice, easy disease. And so I decided that ALS was going to be it, because it had a nice readout of the ALS functional rating scale. And I thought it'd be a piece of cake. Well, it turns out it's not. This is 20 years later. And but we have learned a lot. I think that so inflammation is a normal. It's normal. I mean, we're experiencing it every microsecond. You've got something, a bacteria gets eaten, it causes inflammation, and it recycles back, and it turns off the activation signal. And so the problem that we're having is that in ALS and a lot of these chronic diseases, that somehow this inflammatory signal is not getting turned off.
Right.
And so the question would be, are there different classes of signal that are not being turned off or regulated? And I'm, I did cancer work. I have a lot of my friends. I'm in the hematology and oncology division, and if you think about cancer, there's no one drug for cancer. In fact, people will like throw stuff at me if I said I have a drug for cancer. So I think that we've learned from the sort of the cancer environment and are moving it towards now the ALS environment, and saying that there are factors, whether they be genetic factors, mutations that are passed on in families. I don't know any little children that get born with an SOD1 mutation that have ALS immediately. And so this whole notion about the time wherein something makes you predisposed to a disease, what is going on that leads up to the actual manifestation? So we've really taken a step towards saying we're going to look at the most inflammatory people and learn what's causing those inflammatory changes. And from our experience, I think that almost anything can incite inflammation, whether it be aggregation of proteins, the genetic abnormalities that one sees, misfolded proteins, damage. I mean, soccer players, who knew you get 8 times the frequency of ALS in football players with trauma. So you have these little micro leaks that lead to central nervous system problems. So anyway, I think the approach that we've taken is trying to understand chronic inflammation. And one of the drivers of inflammation chronically in the neurodegenerative disease is microbial translocation of the gut, where you have low levels of endotoxin in the blood in all these diseases, whether it be Parkinson's, Alzheimer's, ALS. And that's a process that keeps this inflammatory loop going. And so we and probably others have been trying to figure out how to put this under better control. And I think that I'm excited. I think we've got a path forward looking at regulation of trying to remove this chronic drive of inflammation. But anyway, so we appreciate that there are different inciters of inflammation, but we're going after what the common features are in inflammation. And I would say that I don't know what the proportion of patients are where inflammation is actually the major driver. I think you have to start somewhere though. So I think that just as a final point, the FDA guidelines came down in 2019 where they acknowledged that ALS is a heterogeneous disease. And if you can take a big group of patients and then focus in on a subset and say, these are people who respond to this approach, and I'm going to design a trial and fix that group. And that's taking a personalized medicine. That's exactly why almost 80% of all the new drugs that are being developed right now are cancer drugs, because that's exactly what people do.
Right.
And so that's, I'm going to leave it there for a time being. I'd love to hear what Rob's working on as far as his the genes and regulation that they're looking at.
Yeah, that's very helpful, Mike. Well, maybe one question before we go to Rob. When you think about neuroimmune mechanisms and the therapeutic approaches, do you see that obviously coexisting with other therapeutic targets and approaches as well? So would it be sort of a foundational approach with respect to the neuroimmune targets and the biology, and then of course there are other factors impinging on that? Is that the way you look at it?
Yeah. So I had the toughest talk I ever gave in my entire life was my daughter's 5th grade class, and I said the brain is like a city, and the houses are the neurons, and the way they talk to each other is the little roads and sidewalks between, and the brain's important because it has these houses. Now, we think that there's a fire that's going on and blocking the ability for people to walk between the houses. And if you want to apply BDNF, growth factors, things like that, you have to stop the fire. And so I think that the approach is very simplistic, of course. Stop the fire using something like our drug, our approach, or any of the anti-inflammatory approaches, and then try to rebuild, because we know that nerves regrow slowly, but they can regrow. So the point is, let's stop something and then build on top of that. So it's not a single answer. It's going to end up being cocktail for sure.
Great. Very, very helpful. Very helpful. Let's go to Rob, actually. And speaking of sort of the next generation targets and the biology, Rob and his team is taking some very innovative approaches with respect to AI/ML, with respect to target validation. So welcome, Rob, and I'd like to hear your perspectives on the next generation of therapeutic approaches in the ALS space. Rob.
Great. Thank you, Murali. I think as we heard from many of our speakers today, ALS is a complex disease. We know that it arises from an interplay of genetic drivers, some of which are known, many of which are still probably unknown, and the interaction of these drivers with a host of other genetic and environmental factors. Looking at individual genes and individual points of therapeutic intervention in isolation may not be providing us with a complete picture of the multitude of pathological processes that are occurring simultaneously within patient cells. An alternative mechanism for discovering new therapeutic targets utilizes an integrative multi-omics approach in which we capture multiple different domains of biological information, such as genetics, transcriptomics, proteomics, and metabolomics, which can improve identification and characterization of pathological processes within disease-relevant cells. Deconvoluting these complex and very large omics data sets requires advanced AI and machine learning techniques to really parse them and plumb them for their deepest meaning. So for example, at Verge Genomics, we've developed a computational engine that can decipher complex omics data and identifies dysregulated networks of genes and proteins that are enriched in disease patients as compared to healthy controls. We can then perform deep bioinformatic analyses on these dysregulated networks, which promotes discovery of new therapeutic targets. And these targets are predicted to broadly impact and reverse multiple pathological processes within this dysregulated network. So beyond the identification of new therapeutic targets, integrative multiomics is also perfectly poised for new biomarker discovery. As proteins or metabolites that are dysregulated in disease can be candidate markers that allow for evaluating the effects of new therapeutic interventions on targets, so target engagement markers, looking for markers, the effects on disease-relevant pathways, and markers of disease processes such as neurodegeneration. So the same AI machine learning techniques that can identify new targets within dysregulated networks are also equally adept at identifying new biomarkers. So as an example of this process, at Verge, we've used this approach to identify a previously unknown pathological process in ALS. So analysis of perturbed gene networks in ALS patients identified a particular enrichment of genes involved in endolysosomal function that were centered around C9orf72 as a genetic driver. Subsequent studies at Verge and follow-on work from multiple other labs has confirmed that C9orf protein, which as Neil mentioned, is downregulated in patients carrying a C9orf72 repeat expansion and represents a loss of function phenomenon. This work has characterized C9orf72 as having a critical role in regulating intracellular membrane vesicle trafficking in endosomes and lysosomes, which impacts multiple different processes in the cell, such as autophagy. At Verge, we identified a therapeutic target which was derived from this dysregulated endolysosomal network. This target is PIK5, which is a phosphoinositide kinase. Inhibiting PIK5 activity can broadly reverse endolysosomal dysfunction, and we have a lead candidate that's advancing towards the clinic. We validated PIK5 as a target using multiple different systems, including human patient-derived systems, such as ALS patient iPSC-derived motor neurons. And we also use mouse pharmacology models of ALS, many of which Fernando's organization works with extensively. We and others are demonstrating that the integrative multiomics approach allows us to decipher complex biological pathways and overlapping pathological mechanism. This then aids in the identification of new therapeutic targets, which we believe will have a greater chance of translating into the clinic and produce disease-modifying effects.
Well, it's very, very helpful, and like the way you laid out, really capturing the disease biology into identifying not only compelling targets, but also biomarkers, which kind of goes back to where Fernando started. How do we really sort out overlapping mechanisms and approaches for combination and coming back to identifying the right patient with the right therapeutic? So it's a step in the right direction, but I want to maybe pause here and have, I don't know if Fernando wants to come back and comment in terms of how do we piece it all together, because right now we have the technology, the way of approaching it that's coming together, along with the aspiration to do some of those studies and put it into practice. Fernando?
Yeah, I think the ALS space is in a much better position than it was years ago, in large part because of a number of sort of collaborative consortia, sort of pre-competitive consortia, a lot in the nonprofit space that have played a pretty big role in collecting large data sets that give opportunities for lots of players in the space to really hone in on patient subsets and try for biomarkers for patient stratification approaches. And so we at ALS TDI, we have a program which we call the Precision Medicine Program, which is rooted in that. It's 8 years running, collecting, it's like a natural history study collected with whole genome sequencing, connected to induced pluripotent stem cell biology, connected to longitudinal blood sampling for biomarker discovery. But we're not the only ones in the space. There's, Target ALS has done similar types of things, another nonprofit. Answer ALS, largely associated with Hopkins, and many other clinics in the United States have done that. I know Canada has recently initiated a similar type of program, and I think we see something similar in Europe as well. And I think more data sharing, more bringing these types of information to the fore for industry and academic researchers will help us get across that finish line in terms of finding the right patients for the right drugs and the right targets.
Mm-hmm. Yeah, absolutely. And following up on that, as those studies get underway, obviously one of the continued questions we wrestle with is endpoints, functional readouts. How precisely can we measure the role of digital approaches? Maybe someone would like to sort of elaborate perhaps a bit more. I don't know whether Michael would like to jump in and sort of elaborate on sort of the landscape with respect to functional endpoints and clinical readouts. Michael?
Yeah. So I think that it's pretty critical to figure out you've identified a subset of ALS patients, and you have a drug that you think does something. I think that a part of what's missing is proof of target engagement. Like, are you actually hitting what you think you're hitting? And that's the strength of TDI. I mean, no question about it. I mean, you guys have screened a ton of different things, and you have a very set series of criteria to say, this is actually happening, and it's a good thing. So one of the things that we've done most recently is looking at very objective measurements of function, in this case, respiratory function, so that you can actually map a single nerve, which would be the phrenic nerve, more or less, looking at respirations using a spirometer, and get hard endpoints that way. So to the extent that you can get a hard endpoint tied with biomarkers, I think that you're really going to move the field forward. And so that's the other thing that Rob's over there. No, whatever, which direction Rob's in. Okay. He's developing his omics approach. I think that that's a very powerful approach. And if you are working with groups that have, and you probably have got your own drugs, just set the bar high about saying, I have to have proof of target engagement using my multi-omics approach before you move to the next step. Because I think that's going to be really a much faster approach to identifying effective therapies.
Great, thanks. Rob, would you like to add anything further from your perspective?
Yeah, I think to expand on what Michael was saying, the deployment of new digital biomarkers, I think, really would be transformational for the way we think about clinical trial endpoints. The example that Michael spoke about of spirometry, which has been, a gold standard in the ALS field. There's some really recent work that suggests that you can get similar type of data with an app on your phone looking at sustained phonation. And it tracks very well with spirometry. And this is looking at this voice analysis can do something at home that you would otherwise have to go into the clinic or work with a skilled technician to accomplish. So I think it's these endpoints that allow us to look more frequently and interact with the patients more frequently, collect more data, really will transform how we think about trial endpoints in the coming years.
Very helpful. Thank you.
Yeah.
Neil, any comments from you to add, Neil?
Just as somebody who's a clinician scientist, at this point, we've come a long way in trial design and in measures of response. I forget who it was who mentioned, but the ALS FRS which used to be regarded as a neither linear nor ordinate, has kind of proven its chaps at this point to be responsive to therapeutics. But it really is the advent of biomarkers. I haven't heard one mentioned yet, neurofilament light, NFL, which also appears to be treatment responsive in motor neuron diseases. So we're at the point where we have a good scale finally, a good clinical scale finally, and we have an existing biomarker which can give us the target engagement or involvement in biology of disease of these various therapeutics. Of course, the future is coming fast, including the digital biomarkers, but at this point, we have these two useful measures of response.
Thanks, Neil. That's great. So for the remaining time, I'm going to assume that I'm an ALS patient, and I'm going to ask the question, what can I expect in the next 5 years? What is your vision for how the pipeline or the therapeutic pipeline is progressing? And with a view from the patient, what can I expect for a viable treatment? What are the mechanisms that you like, for example, or which are the ones that are progressed further along? It will be helpful to get some guidance and insights from each and every one of you. So I'm going to maybe start with Fernando, who is actually on my left. Maybe start from Fernando, and then we'll go through each one of you. Fernando.
Sure. I think at the top level, we can hope and expect a few new approvals that are already in the pipeline. There are a few things that are under FDA review right now, which have shown some promise clinically. And we can hope for new options to be available for people right now. We don't expect that those options are going to be transformative, right? I think what's in the pipeline right now there are some therapeutic approaches that have potential for profound effect on some people. And there are some therapeutic approaches that have potential for a good effect on many. But we don't have many therapeutic approaches that have potential for a profound effect on all. And so I hope that that we can strive toward sort of that clinical toolkit that can get eventually to profound effects for all. But it might be a complex cocktail, and it might be different for one patient to another. So I think an ALS patient might expect for more sampling to happen in them, right? So over time, we may end up seeing more genetics play a role in their diagnosis and prognosis, and then therapeutic targeting, and maybe assessment of biomarkers like neurofilament that Neil alluded to, seeing if you can get a prognosis on disease progression, and then bending that prognostic curve for each of those people with drugs in the clinic. So it's a better time than ever, but not good enough.
Great. Well said, Fernando. Maybe pose the same question to maybe Rob.
All right. Maybe take a slightly different approach. And I think one of the more exciting developments in the past few years is the emergence of platform trials and other large research groups and initiatives that are really going to allow us to centralize a lot of the information that we receive from trials. The Healy Platform Trial Group in Europe, we have NCALs and TriCALs. I think these groups are going to be transformative to how we think about data collection in our trials. And they also represent an advantage for patients in that the structure of at least the Healy platform is that you have a significantly greater chance of going on to a therapy versus being in a placebo group, and the ability to enroll in multiple trials. And I think that's going to represent a great advance for patients.
Thanks, Rob. Maybe pose the same question to Neil, and then we'll go to Mike. Neil?
Sure. So I agree with my colleagues, but I heard Fernando use the word hope. And this is what I convey to my patients. I've been in the field for almost 40 years, and there's never been a time in history when more people are working on ALS and more companies are in the space for the purpose of developing and testing drugs. So this is, as Fernando said, there are 2 compounds right now under consideration at the FDA. That's also been something that I've never experienced. So there's a profusion of new approaches. There's an incredible amount of activity and funding. I think that the prognosis for the next 5 years is very good. So I think we're in for something good in ALS.
It's very hopeful. Thank you. Mike, do you want the last comments here on this topic?
First of all, I think that it's incumbent upon us to figure out how best to use ALS patients' very valuable but short period of time where they can actually be in a clinical trial. And that's why I think that moving towards more of a little bit like a cancer approach and say, all right, what's an acceptable response rate, whatever that response is, and define it with some kind of an objective measurement. In cancer, you look at, did it shrink or not? Well, we're taking a look at this whole spirometer kind of a thing, just saying we're looking at one lower motor neuron endpoint, and that is the functional respiratory function. That plus biomarkers tracking along together, because I think that it's important that you don't do a clinical trial where somebody occupies 6 months to a year of their life. And I think that Rob's comment about the Healy platform, I think the platform is spectacular. Great idea. I would also like to encourage there to be other sort of unique approaches, the kinds of things that Fernando's been doing, but have short-term biomarkers as a readout and tell the patients to say, you know what, you're participating in early steps of something, and we need you for a month or something like that because we want to try to validate a target. It won't keep you from being in other trials, but I think that with this new way of looking at digital biomarkers, things like that, we've got to accelerate the endpoint readouts so as to encourage pushing some of these things through across the finish line.
Great. Well, these have been very, very insightful comments today. We heard about how emerging biology is being tapped into with new approaches, the multiomics, the AI/ML approaches. We heard about commitment from not only companies but also foundations and other organizations. The need for increased collaboration in this space. So overall, this has been a very illuminating webinar. I really appreciate and thank the panelists for joining today for this conversation. And I hope those of you are watching it do really enjoy this discussion as well. With that, we'll bring this to a close. Thank you.
Thank you, Murali.
Thank you.
Thanks to our deeper understanding of disease biology and technological breakthroughs, we are seeing a new wave of innovative therapies across different modalities. Yet we need to make more progress to be able to achieve the goal of delivering precision health solutions to treat ALS. What are the key elements to deliver the promise of these new modalities as transformative medicines? What are the key roadblocks and excitements ahead? In the next discussion, our panel speakers will share their insights about challenges and opportunities in new modalities. Please welcome Jim Burns, CEO, Volcano Bio, Clive Svensson, Professor and Executive Director, Board of Governors Regenerative Medicine Institute, and Yibhash Mokapata, Managing Partner, Sound BioVentures.
Well, thank you to Wuxi for hosting us today. And in this panel, we talk about new modalities for ALS and neuromuscular disease therapy. We have two very eminent panelists who approach the world from different viewpoints, given their seats and vantage point. So far in the conference, you have heard about or will hear about the broader trends in ALS granular understanding of the disease biology, new therapies that are coming online, and very importantly, translational work that's going on in as a general direction to better understand disease, ultimately, so that patients can benefit out of the new therapies that are being discovered. So with this specific panel, what we will do is pick the brains of Jim Burns and Clive on the new modalities of therapies that are coming on. What do we mean by new modalities of therapies? We will primarily touch on cell therapy, gene therapy and editing, and RNA-based therapies. And this, as you can understand, historically, most of the therapies have either focused on small molecule approaches or antibodies and so on. But this is a new, as with many diseases, ALS too, is getting to a point where these new modalities of therapies are being discovered and being developed. Let's start with introductions. Jim, maybe you can give a quick intro about yourself and your professional background for the benefit of the audience, followed by Clive.
Yeah, thanks, Bash. I appreciate that. And first of all, I'm also really happy to be here. I think this is such an important topic. And, you know, I'm the CEO at Locana Bio, and we're really excited about the approaches that we're taking for trying to address a familial form of ALS. But before I get to that, you know, I've been the CEO of Locana Bio for a little bit over 2.5 years, and the company is a private company, and it was the brainchild of Gene Yao over at UCSD, right down the street from where I'm standing right now. And he had the concept of actually trying to target dysfunctional RNA with naturally occurring payloads, such as RNA binding proteins that could be designed and engineered to target the disease-specific RNA molecules that could be, or RNA, which is ultimately could be toxic in causing the disease. So what we're doing is engineering those for a number of different applications. And in the neuromuscular space, in neurodegenerative diseases, that's where we're starting. And what's different about the approach that we're taking is that we're also delivering these by gene therapy. So our premise here is to be allele selective for the disease-causing RNA, spare the wild type where we can do that, to be able to have a durable response with a single administration via the gene therapy. And there's another very important feature of this is the ability to do what we call multi-targeting, which I'll get back to, which we're also really excited about. We're a 75 employee company at this point in time. I mentioned that we're a private company, and we're at this stage where we're generating leads and advancing towards IND-enabling studies in the neuromuscular and neurodegenerative area.
Thank you. Clive?
I direct the Board of Governors Regenerative Medicine Institute at Cedars-Sinai in Los Angeles. Yeah, very interested to be in this session, and I think complementary to what you just heard from Jim, our approach has been more along the lines of sporadic ALS. Now, what we do know in ALS is that the motor neurons obviously die. That's what causes the disease. There's a feeling now that these neurons die in part because they lose support cells called astrocytes. Now, astrocytes are very important in the brain. They actually control the blood-brain barrier. They protect the motor neurons. So we've really been focusing on a combined stem and gene therapy approach to replace the astrocytes in ALS patients. We derived a way of growing fetal-derived astrocytes from fetal tissue, fetal brain, but they didn't have a large protective effect on the motor neurons. We since found that actually there's another factor called GDNF, a growth factor, which is known to protect motor neurons from death. So when we combine those two things, GDNF doesn't get into the brain or spinal cord, it doesn't get across the blood-brain barrier. So we thought, could we use the astrocytes as a Trojan horse, transplant the astrocyte precursor, but engineer it using gene therapy, it's called ex vivo gene therapy, to produce GDNF. We've got them into patients, we've shown they can make cells that look like astrocytes, and they'll secrete the GDNF over up to a 4-year period in the spinal cord of patients. So we're very excited about that. We're now moving to the motor cortex. In fact, we're doing our first patient in about 3 weeks. Very quickly, in vitro, because we don't know the cause of ALS, we're developing really cool models. There's this technology called induced pluripotent stem cells. And together with Answer ALS, we created 1,000 induced pluripotent stem cell lines from patients with ALS. Now, these cells, once you've made them, iPS cells, you can make them into motor neurons that die. It's a fascinating resource. We hope by doing that, we can make the motor neurons in the dish. We may be able to understand, we can sort of model the process of the motor neuron dying. We may be able to get new targets so that Jim can develop new gene therapy approaches or ASOs for those new targets. So that kind of technology is coming along nicely, and we can talk about that if you like as well. That's my summary.
Perfect, Clive. Let's actually build on that, because what's very evident is just given the differences, right, in overall understanding of the human genetic, well-defined in genetically defined versus not so much in sporadic, and the patient populations being sort of very different, yet the clinical presentations in many instances having common themes. The iPSC differentiation approach holds enormous promise. So maybe you can elaborate on what the overall promise there is and what the key elements are for those that approach translating into a real drug.
Yeah, I think the first stage is to show that you've got a phenotype in your motor neurons. So we can make the motor neurons, but the whole field now is waiting with bated breath to know whether or not an ALS motor neuron is different from a control motor neuron from a patient who doesn't have ALS. And it's turned out to be an enormously complex question, mainly because of the variation. When you differentiate, you turn iPS cells into motor neurons. There's quite a lot of differences, both every time you run the assay, you get slightly different numbers of motor neurons and astrocytes and other cell types, and patient-to-patient variation is larger than we expected. So when you take into account the variation, to see anything related to the disease becomes very tricky and harder. Having said that, because we have such large numbers now with Answer ALS up to 1,000 lines, we're starting to now tease out subgroups of ALS types that then will differ from control. And that's the goal of Answer ALS. And if you can find those, say, 4 subgroups that have a particular molecular signature, then you could go target some drugs towards them. We're not there yet. We're still analyzing this large data set. And we're actually, it's an open source system, Answer ALS. If you look it up online, there's a portal you can go in, and anybody can look at the data and try and see if they can find targets within our data. We do RNA-seq, we do proteomics, ATAC-seq, and whole genome sequencing. And it's all in one place. You can correlate that with clinical outcomes because we've got all the clinical data. So we're hoping that we may not get the answers, but others might use this resource to find the answer and find the subtypes again. So Jim and his company can have some more of the targets that would work. But we're not there yet. It's a very hard task, actually. And IPS technology is complicated, but we're very excited at the possibility. I think IPS are going to replace or complement animal models in the future and be an essential part of drug discovery.
Could I just comment?
Please.
So I 100% agree with that last statement. And that's part of our screening cascade. In fact, we think it's almost more important for us to be able to show that our constructs are having activity in more of a disease environment with patient cells and than in the animal models, although they're very complementary. But I can't resist asking the question: do you think that there is a difference that between the lower and the upper motor neurons that you're creating with the iPSCs?
So again, so just to go back to the C9orf, there are specific changes that we see for genetic forms of when you make IPS from C9 patients, you can see specific kind of pathways, and you see dipeptide repeats spitting out, so you can see phenotypes. With sporadic, we're mainly focusing, to answer your question, on spinal cord motor neurons. We kind of ventralize them with sonic hedgehog and make them into, but complementary, we're now doing the upper motor neurons and upper motor neurons and the cortical cultures. The answer is still no. We haven't got, from our lab anyway, a specific phenotype that would say, if I look at that culture, I know it's an ALS one versus a control from sporadic patients. It's just too much variability at the moment. But I'm telling you, we've got a lot of data going on. There's lots of groups around the world really focusing on this. I think the answers are going to come pretty soon as we get these targets. And we just don't have enough data yet, Jim, to really nail it.
Yeah.
The critical bit there is this translation using human samples, right? And going iteratively between that and a ex vivo model, and then being able to tweak it to the best of biology so that it can mimic or replicate the disease pathology. One thing I must highlight, which actually I've underappreciated, is this quote unquote distributed nature of the processing, right? It's very, very important. As you said, Answer ALS, people can go in and use their resources to find it. And this is kind of a resource for patients ultimately, right? As well as for researchers. And that distributed processing is something that doesn't happen in many diseases, but in many orphan diseases it does. And when it does, it becomes extremely powerful. So kudos for getting that. So effectively then, Clive, what you're saying is that there is an exercise with large translational data sets to replicate and understand the pathology that happens in sporadic ALS. And that's very important because what you couldn't determine by genetic backup, you would otherwise dump into the bucket of sporadic ALS. But with time, a pattern will begin to emerge where sporadic ALS also becomes understood with regards to molecular pathology.
Yeah, this is where we hope to be. So when Jim does his trial and he's enrolling patients, we'd like to say you'll get the genome from the patient, obviously, so we know if they're C9, if they're SOD1 and other mutations. The next level of that is going to be you'll get the genome, and we know you can't really distinguish ALS from control just from the genome. I mean, there are PRS scores, polygenic risk scores coming, but it's not definitive. Mm-hmm. I think the next level will be from his 10 patients in a small trial, you'll make IPS lines and you'll make their either cortical neurons or motor neurons, and you'll be able to select the patients that you predict will work because you'll be able to try your drug, like Jim just said, on those lines before you enroll the patient into the trial. That's the next generation of clinical trials that we've got to get to. We're a long way off that, but I think all of us are working to that precision kind of health idea in ALS, and not just bucket everybody, because that's what we do now. If their vital capacity is this and that, their exclusion criteria, we're doing that right now for a trial. We've got to be more sophisticated, right? Because that's just not where we are with the molecular techniques that we have. So that's the goal in the future.
So Jim, building up on that then, can you help the audience walk through what it would be like, sort of gazing into the future with this sort of a data set that Clive has? Let's assume that one or two patterns begin to emerge. How will that feed into the discovery approach that you, Lokana, and the field in general can take?
Well, I think what's really important here is that, as Clive is saying, is that the ability to really understand what mutation or sets of mutations does a patient really have, because then you have the best chance of actually answering the question about whether or not it deserves to, or they should be getting the product that you're going to be testing in the clinical trial be most meaningful for the patient. And what we're doing, and Clive, I'm going to guess, and I think this is maybe, just thinking 10, 20 years in advance, you're going to discover through your work and others that sporadic ALS, that there are mutations that are causing what appears to be sporadic, and that maybe it's like what oncology has become, or is becoming, where it's a host of rare genetic diseases. And the more we can understand that, the more likely we are going to be able to offer hope to patients, because we understand more of the molecular basis of the disease. What we're doing, I think, which is interesting, because we understand to a certain degree the genetics, that for C9orf72, it's this hexanucleotide repeat expansion in that gene. And once it gets beyond a certain number of repeats, the transcribed product of that, the RNA, becomes toxic. But what's interesting about this, and Clive, you do a better job than I can in describing this, it's the sense as well as the antisense that transcript that is toxic. In addition to that, the dipeptide repeats that you mentioned as well. So what you need to do in that case is understand that the genetics of that, and be able to construct something that can actually, in a single product, this is what we're trying to do, either destroy or somehow block those long repeats so that they no longer sequester binding factors, and so they no longer make those dipeptide repeats. And unless you understand the molecular basis of that, you would maybe just go after the sense versus the antisense, but you go after both. Ideally, wouldn't it be great, Clive, if you could also, because this I think it's controversial, but it seems like maybe this is also a haploinsufficiency disease as well. And if that's the case, could you also do something to try to upregulate the transcription of the normal gene? So by knowing more about the molecular basis of this very complex disease, you have a much better chance of designing a product ultimately that's going to benefit patients. So that's my hope is down the road, is that we have multiple ways of trying to address this for patients because we understand more of the molecular basis of the disease.
Mm-hmm. Can I ask an orthogonal question here? So understanding the basis of the disease is absolutely critical. Does it also shine light into the time of intervention or the extent to which one can reverse an already existing pathology using these modalities?
I think it comes back to the modeling, and I didn't bring it up, but and Jim, whether we need more sophisticated models is something we talk about a lot. One of the downsides to the IPS modeling right now is we don't see overt death of the motor neurons, right? They change, they have phenotypic changes. We'd love to see death because that's what happens in ALS. We know that's the endpoint. And if you can reverse death, then you've got a product which is, so we're kind of fighting to get the sophistication of the model. And so to your question, Vibash, if we could longitudinally show, okay, this is an early stage and then the neuron pops and we can sort of intercede in our model at any point with our drug, that's fantastic, because then you can ask that question, when should you be giving it?
Right.
We're trying to develop these organ-on-chip models. Now, these are really interesting because there's a little microfluidic device, and on the top you put your neurons, you differentiate your neurons out from the IPS cells. And on the bottom, from the same patient, we can make endothelial cells, or BMECs, which are the blood-brain barrier, basically, the blood vessels that line the brain. So then you can give your ASO either on top direct, which is sort of artificial, it's like giving it through the CSF a little bit, or you can give it through the blood, which ASOs don't penetrate very well through the blood, so you normally would give it on the brain side. But then you can give it at different stages of the disease process. So if we can get this model more maturing and we actually see the cells dying in the dish through some stresses, I think that's the ultimate model. And you could ask the question you just asked, and Jim can say, we can only take patients at this stage of the disease, because we know when we add the ASOs at this point, they don't work anymore. Right. So it definitely will validate a lot. Now, there are mouse models, but as you know, the C9 mouse model arena is very complex, and there hasn't been really reliable mouse models in that. We all rely on the SOD1 still, more or less. And so, these human models, I think, will make a difference. But the last thing I'll say on this is we have to get validation. I mean, I'm excited about the models, but we need to validate the IPS models across just something. Now, the problem is there's nothing that works very well in ALS. So what are we going to validate it on?
Yeah, the other part of that is, in terms of thinking about when you treat patients, is that as we think about how we would trial what we're developing, I mean, naturally you go into patients that have already been established to have the disease, and the question is it too late for some of these patients?
So has there been approaches which basically can not only prevent the death or future death of neurons, and keep them alive, but also regenerate neurons in the right kind of neurons? Is that a possibility that's relatively soon on the horizon? Is it even doable scientifically?
Well, I can go first because this is quite close to my heart, having done regenerative medicine for 30 years now. And the one thing we know we can do in Parkinson's is replace dopamine neurons. So you can take fetal tissue or even iPS cells, make them into dopamine neurons, progenitors, put them in the striatum, and they survive, they make dopamine. The thing that we're not very good at is recreating the circuits after we put these neurons back in. Now, with motor neuron diseases, neuromuscular diseases, the distance is so massive. If I can make a motor neuron, actually, I could even put it into your brain. I could even put it in the right place. The problem is, how do you get it to reconnect?
Right.
It's got to go 3.5 foot down. Now, all your neurons were born before you were. And most of them were connected before you were born. In other words, you know, neurodevelopment is amazing. It's everything connects up and then you've got to stretch. So we're asking the impossible almost to get in a human brain, the upper motor neuron to reconnect out. So I feel like that's true, obviously, for a lot of different neural circuits. So making interneurons may be different, you know, short neurons, but I think we're really tasked in motor neuron diseases, which is why we went the astrocyte route. Let's protect the neurons that you've got. I think that's more likely to work in my lifetime. Once someone's clever enough to generate a motor neuron, maybe there's a fiber tract that it can find and put an axon all the way down. It's been hard to do that in mice even. So, but boy, I'd love to be able to do it. I think regenerative medicine technology is moving in the future. My graduate students, hopefully in 40 years, will be doing it, but it's hard right now.
Right. No, understood. Pointing on something that you said, Clive, about lessons from broader neuromuscular disorders, are there certain themes that are cross-pollinated between ALS and broader neuromuscular disorders that we can learn from, either in terms of translational work or in terms of regulatory lessons learned and so on?
I think the big, you know, the elephant in the room is spinal muscular atrophy, which we haven't talked about. And that's one of the most interesting childhood diseases, because there are actually 3 different therapies for that disease right now that have been approved. And that's so rare for any neurological disease. And I don't know why it happened for SMA, but it certainly did. And SMA is caused by single mutation, SMN gene. So we know the mutation. That helped a lot. It's early in development. And I think the gene therapy approach, and I'll ask Jim for his thoughts here, was done now, you know, now it's Novartis that are leading this. The gene therapy approach was pioneered by Brian Caspar, is simply just to put the gene back, the missing protein. And I think the reason it works so well is because it's a childhood disease. So when they gave the product interest ICV, it could distribute to large parts of the spinal cord and brain in these kids, and they got nice functional effects. It goes back to now doing that same thing in an adult neurological disease is a bit more tricky because everything's a lot bigger and it's just distribution and delivery might be a problem. But it definitely shows us that it could work, that gene therapy works. And these kids are really remarkably better. They're not cured completely, but they're a lot better. Then came a drug from Roche that seemed to work pretty well, and that's already approved. And finally, an ASO, and Jim might want to talk about that, an ASO for exon skipping from Ionis and then Biogen. Amazing. I mean, that's in the market as well. So, you know, it's just funny. And it's like in England with you know, you're waiting for the number 3 bus and never arrives, and then 3 come at once. And I think it's a bit like that for SMA.
Yeah, and what I think is really interesting about that story is that the ASO, I believe, was the first product, and you know, just showed tremendous promise. What a great thing for the patients. And I think that's what we all aspire to in these diseases, is to be able to give patients multiple options. You know, all boats rise when ASOs work or gene therapy works, and you know, it's giving patients more hope and it's giving them more options, and there's plenty of room for products in this space. And you know, I think there's a lot of learnings that you know, as we think about what we're doing for C9, which is a repeat expansion disease, and thinking about DM1, which is a neuromuscular disease, obviously, which is also repeat expansion diseases. There's crosstalk there in terms of your learnings and how you think about how you're going to block or destroy those repeat expansions and spare the wild type. It even goes for, you know, for Huntington's disease as well, which is a neurodegenerative disease, you know, very similar types of learnings, a lot of differences as well. But I think this is one comment, which is that there's so many of us that are working in this space. Wouldn't it be great if in a pre-competitive way, we can make sure that the learnings that are helping accelerating all of us to develop products that will help patients, you know, in an ideal world, to be something that we'd be doing a lot of. And I think with natural history studies, that's something that does happen, and that's a real positive. So Anyway, I think it's a really exciting time if you're in the genetic medicine space and you're thinking about what you can do for these different types of diseases that we're discussing.
Fantastic. So, gentlemen, keeping in mind the time, maybe we can wrap up with 2 questions rolled in one. This is a gazing at an 8-ball, looking out into the future kind of question. So, from your vantage point, and having each of you having been in the space for decades and seen the space evolve, what would you like to see in the next 2-3 year window and then in the next decade window that ultimately you think would be absolutely critical for not only intervening, but ideally curing patients of ALS? Who wants to go first? Clive?
Jim, go first. I'll be polite.
All right. Well, I'm going to lean on Clive a little bit. I want more genetic mutations to work on. So I think truthfully, I think that's going to make a difference for patients. But the other is, quite frankly, it's delivery. The payload is really tough. Understanding the biology around that. And let's say that you had the perfect knowledge of what that is. Delivery is still really difficult. And, so I think, in our case, we're using viral vectors. I could envision sometime down the road, you have non-viral vectors. But I think what makes this this area really interesting in the future is if we have improved ways of delivering either systemically or locally to get better targeting to the areas where you need to get to, whether it's to the motor neurons or in the case of FTD, right? Because C9orf72 also is involved with the familial FTD, how you distribute it more broadly within the brain. So I think that's a big challenge for us in the future.
Yeah, and I'll double down on that, Jim, because we're doing our first patient very soon. We're actually delivering cells to the motor cortex. And that paradigm could be used to deliver drug, AAVs or even ASOs directly into certain specific regions. Because one of the problems with gene therapy is the off-target effects, right? And it's the same with CRISPR. It's that other elephant in the room, which is off-target. Yeah. So I think if we can do more trials innovatively with neurosurgeons love to put stuff back. They're used to taking stuff out, but they can inject a drug or a gene or something, it's possible. And I think people don't go that delivery route. The nice thing with that is you limit the off-target effects, because if you just put in one part of the brain, that's where the drug is. You can actually look at that with imaging as a biomarker and focus on that area in a patient. And these ALS patients really want you to try these innovative approaches. There are very innovative ways we can progress with small trials, with new gene constructs across many patients. And I think we should be more innovative over that and not so scared to it's brain surgery, but it's actually the only way we can understand more about the disease. And the last thing I'll say is these platform trials that MGH is running now that they've used in cancer, I think are going to enable us to look at more products faster, because this old trial design is just not you doing it for ALS. We need to go into the cancer model, go for platform trials. That's where you have 10, ideally 10 different drugs with one placebo group that covers all of those different patient groups. And I think if we could get more inventive with those and include some of Jim's kind of trials, the ASO trials and gene therapy trials in platform, and this gets into the pre-competitive space, all working together, I think we can accelerate things much faster as a team than trying to do these individually and raise 10 million. And we should all be working together. This is about ALS and a disease that's the most horrific disease you could ever be diagnosed with. And pre-competitive spaces, you mentioned, Jim, that's where we could all work together and just share results, share prelim data. Let's get this on for the patients, not for us. All right. Perfect. I'll stop there. I'm getting on a soap thing, so I'll stop.
But that's perfect. And on that note, it's basically boils down to right gene, right amount, right place, and right time. And on one hand, and then knowing the granular detail, the underlying pathology, and the molecular intervention we need to do, that will drive ultimately new discoveries and new drugs. This has been a fantastic conversation, gentlemen. Thank you so much for participating, and onward and upward.
Thank you. All right.
genomics, and big data are transforming how we treat numerous conditions. How can we integrate these into drug development to combat challenging diseases like ALS and neuromuscular disorders? Please welcome Michael Snyder, Professor and Chair, Department of Genetics, Director, Center for Genomics and Personalized Medicine, Stanford University.
We're in an incredibly exciting time in science now. You can collect data at a level that's never been possible. For example, we can sequence people's DNA. We can profile thousands of molecules out of people's blood and urine. We can collect information about their physiology using wearable devices. And in fact, I use 8 of these devices every day. You can see my 4 smartwatches here. Why are we doing all this? Well, by collecting data, we hope to understand what people's personal baselines look like and then detect disease early. So for example, we can see if you're getting ill by shifts in your patterns, either your molecular patterns, your changes in physiology from wearables. We also think genome sequencing is going to be super important for risk prediction. And in fact, we just had a recent paper out on ALS, finding 690 genes that are responsible, that seem to be responsible for some of the genetic underpinnings of the disease. And hopefully we'll turn that into risk predictions, meaning just from a genome sequencing, genome sequence, we can predict who's at risk for ALS and who's not. And ultimately, I think all this data is going to be very, very powerful for, again, doing risk prediction, following people's health, and catching disease early. So genome sequencing itself is going to be very, very powerful. We're not too far away from the $100 genome, where everyone will be able to get their genomes sequenced. And then together with these new methods for disease risk prediction, a lot of which use artificial intelligence, machine learning, we'll be able to actually predict better who's at high risk for the disease. The challenges are to be able to make sure we can figure out the right treatment for the right person. Right now, a lot of that's trial and error, and I hope we'll be better predictive in the future. Complex diseases are rarely a single size fits all. Usually there are a multitude of diseases. Something like ALS probably involves several different forms of the disease, and different treatments are likely to work better for different people. And so we're in a world where we can probably figure this out now much better. At least we can see who's responding. We need to couple this with valuable methods for making new drugs to combat these chronic diseases, these very problematic diseases like ALS. And I think we're in a world where it's now much faster to be able to develop drugs. So we hope that this will accelerate the whole science as well. So I think the combination of using big data to be able to, again, manage people's health, try and keep people healthier, in combination with our new technologies for being able to better design new drugs. And I think data is also going to be very valuable for being able to follow whether treatments are working or not. If we can profile people at a much higher level, we'll be able to tell, who's improving, who's not. I think the other reason this is all very important is that a lot of this needs to be done at the personal level. So rather than just trying to do one treatment for everybody, I think in the future, we're going to discover that complex diseases like ALS, they're really a multitude of diseases, and actually different people will respond differently to different treatments. And we can follow this for the first time using these deep data profiles on people. One of the challenges is getting the right markers for these different forms of the disease. I think this is where data-driven medicine will be very, very powerful because we'll be able to see who's better responding and who's not if we have better measures of people's physiology. And again, I think big data and wearable devices are going to be very, very powerful for that. I think we're still, we're better at making drugs. I think, though, getting the right drugs for the right person, that is still going to take quite a bit of effort. But I think we are much faster at making new drugs. I think the awareness actually of ALS and the fact that it is such a debilitating disease is going to help drive a lot of the industry towards trying to make better drugs and hopefully more effective drugs. We're also in a world where you can design drugs better using molecular modeling systems that hasn't been possible before. And I think that should also help us quite a bit. So I'm very optimistic that data-driven medicine will be powerful for keeping people healthy, catch disease early, and then ideally being able to treat disease at a personal level that's never been possible before. And ultimately, though, I think the combination of all these things, the genetics, other forms of data collection, and personal drug design could be very, very powerful for actually getting more effective treatments, again, for these very debilitating diseases. And so I think we're in for a very bright future, and it's all going to be driven by big data.
Please welcome Richard Saul, Head of Boston Office and Senior Advisor of Strategic Initiatives, WuXi AppTec, who will close out our event.
Neuromuscular disorders typified by ALS are enormously complex diseases. Yet we're encouraged by the continuous insights revealed by advances in science and technologies, with the hope that these, coupled to strong patient advocacy, will lead to better diagnosis and care, as well as to herald in a new era of therapeutics for ALS and neuromuscular disorders that will transform the lives of these patients. Through collaboration, we will more effectively tackle challenges and lower the barrier towards innovative solutions for patients. We look forward with great anticipation to the fruits of today's discussion. On behalf of the WuXi AppTec organization, I'd like to express my gratitude to the distinguished speakers for all of their remarkable insights, to the ALS Association, to the Muscular Dystrophy Association, Motor Neuron Disease Association Singapore, Beijing Oriental Rain, ALS MND Care Center, and many other global partners. Thank you so much for your support. And thank you to our audience for joining and participating from all over the world. At WuXi AppTec, we believe in a future where every drug can be made and every disease can be treated. With over 7,000 rare diseases and only 5% available treatments, there's no question that this future is possible only if every one of us works closely together. Thanks again for your participation, and we hope to see you at the next episode of our Collaborations That Transform series. Thank you very much.